After transcription, the eukaryotic pre-mRNA, also known as the primary transcript, needs to be processed in various ways. These processing events include 5' capping, splicing, and polyadenylation. In 5' capping, a modified residue called 7-methylguanosine cap is added at the 5' end. In splicing, introns, which are non-coding sequences, are removed, and exons, which are the coding sequences, are joined together to form a continuous sequence that specifies a functional polypeptide. At 3' end, adenine residues are added to create a poly-A tail. All these modifications leads to the formation of a mature mRNA that can be exported from the nucleus to carry out the translation process. In this video, we will discuss in detail about the 5' capping process. The enzyme involved in eukaryotic transcription is RNA polymerase II. It has a carboxyl terminal domain. It is believed phosphorylation of serine residues in carboxyl terminal domain serves as a platform for recruitment of RNA processing factors. Phosphorylation of serine at position 5 is associated with the recruitment of capping factors. Modification at the 5' end of the primary transcript occur at very early stage of mRNA synthesis, just after the first 20 to 40 nucleotides of the transcript have been added. The capping enzymes are recruited by the carboxyl terminal domain of the polymerase enzyme. It involves phosphohydrolase, which is also known as RNA triphosphatase, guanylyl transferase, and methyl transferase. All the three of the capping enzyme and through them the 5' end of the transcript itself are associated with the RNA polymerase II carboxyl terminal domain until the cap is synthesized. The 5' ends of the mRNA initially possesses a triphosphate. In the first step, the last of the 3-phosphate is removed by the enzyme RNA triphosphatase, converting the 5' terminus to a diphosphate. The next step, the enzyme guanylyl transferase adds guanosine monophosphate residue in an inverted orientation to form an unusual 5' triphosphate bridge. In the next step, guanine 7-methyltransferase adds a methyl group to the 7th position of the guanosine base to form 7-methylguanosine cap. The methyl groups are derived from S-adenosylmethionine. Let's discuss the capping procedure in a more elaborate way. The enzyme RNA triphosphatase removes the gamma phosphate at the 5' end of the mRNA. The enzyme guanylyl transferase catalyzes the nucleophilic attack of the beta phosphate on the alpha phosphoryl group of a molecule of GTP. The beta and gamma phosphates of GTP serves as a pyrophosphate leaving group. The guanylyl transferase adds the GMP in an inverted orientation so that the 5' end of the guanosine is facing the 5' end of the mRNA chain. As a result, the first two nucleosides are joined by an unusual 5' triphosphate bridge. If we see the structure in this way, GMP is added in an inverted orientation to form a 5' triphosphate bridge. In the next step, the enzyme guanine 7-methyltransferase adds a methyl group at the 7th position of the guanine base to form the cap 0 structure. The enzyme 2-methyltransferase often adds additional methyl groups to the first and the second nucleotides adjacent to the cap. These methyl groups are added to the second position of ribose sugar. If it is added to the first nucleotide, it forms the cap 1 structure. If methyl group is added to the second nucleotide as well, then it forms the cap 2 structure. The methyl groups are derived from S-adenosylmethionine. After capping, dephosphorylation of the serine 5 within the carboxyl terminal tail leads to the dissociation of the capping machinery. After the capped 5' end is released from the capping enzymes, it binds to the cap binding complex. Cap binding complex is composed of proteins and participates in binding of the mRNA to the ribosome to initiate translation. The 7-methyl guanosine cap has several important roles, such as protection from degradation. The 5' cap protects the mRNA from degradation by exonucleases, facilitation of ribosome binding. The cap binding complex binds to the 5' cap and helps the mRNA to bind to the ribosome for translation initiation. Nuclear export. The 5' cap is recognized by the nuclear export machinery, facilitating the transport of the mRNA from the nucleus to the cytoplasm. Regulation of splicing and processing. The 5' cap interacts with other proteins and RNA elements that regulate pre-mRNA splicing and processing, ensuring the proper maturation of the mRNA transcript. Let's discuss some objective questions related to the topic. Which of the following is not the step of mRNA processing? RNA silencing. Which of the following forms the 5' cap? 5' triphosphate linkage. During capping, if methyl group is added to the guanine residue, it is known as cap 0. Which base is present in the 5' cap? Guanine. What modified base is at the 5' extremity of a capped eukaryotic mRNA? 7-methyl guanosine. What is the role of 5' cap? Protection from degradation, facilitates binding to ribosome, nuclear export. All the above. The enzyme used for capping mRNA is transferase. The enzyme not involved in 5' capping, topoisomerase. The 5' end of mRNA initially possesses a triphosphate.